Welcome to the ASEF podcast. My name is Tanja Janko, ASEF Fellow 2019. I studied chemistry at the University of Zurich and chemical technology at the University of Ljubljana. And I am hosting the Science Vertical, where we will navigate diverse knowledge landscapes from the depths of quantum physics to the intricacies of social dynamics. Together, we will uncover the mysteries, discover the current state of research, and discuss the challenges that shape these fields. Today, we will be talking about computational biology, and it is my pleasure to introduce an expert in the field, Peter Cimermancic, who is a co-founder of TesorAI and has extensive experience in computational and structural biology, genomics, proteomics, and target discovery and modulation. Peter, welcome. As a kick-off question, I'd like to ask you what initially drew you to the field that you're currently in, and what continues to motivate you to do your research?
Well, first, thank you for having me. Pleasure to be chatting with you. And hopefully our conversation is going to be helpful to at least some students who are thinking about, let's say, the same type of problem as we both are. But yeah, to answer your question, since being a kid, I was always very curious about how different things in nature work, how, let's say, the processes evolve. Drug discovery was something that I've been drawn to quite a bit, especially being, let's say, from Dolenska Novo Mesto, having Krka factory nearby. That was something that I was always quite excited about. In college, I know I tried wet lab, and that just wasn't a good fit for me. I guess I don't have the right personality. I have much respect for people doing wet lab, but for me, it was perhaps too slow. Experiments are not, or when things go wrong, certain factors are not always in your control. So it's difficult to, let's say, troubleshoot what went wrong, and things take some time. So I was very glad to get introduced to computational aspects towards the end of college, because that seemed to be a lot better match for how I like to work. I just enjoyed that intersection of math, logics, let's say, being able to asking questions and getting responses, answers quickly, you're in control. So if something goes wrong, you're likely responsible for introducing bug in your code and stuff like that. And let's say in today's day and age, where a lot of biology is hypothesis free, or we are, let's say, generating data in more fishing expedition type of an approach, like just knowing how to ask questions and get the answers back from that type of data, that's just something that I'm super excited about. So that's what motivates me about doing research in computational biology.
So Peter started his studies at the University of Ljubljana, where he studied biochemistry. And I read in one of your interviews that you visited Professor Andrei Shali at UCFS, still during your bachelor studies. And after that experience, you decided to pursue PhD at UCFS. To me, this is truly the essence of what we want to bring to our future ASEF students. So could you please tell us a little bit more about that experience, and also how would you compare your experience studying in Slovenia versus studying in US?
While in Slovenia, I was working with Professor Brigita Lenarčič quite a bit, and she introduced me to Andrei at UCSF, just took the opportunity to spend 5 months in my last year of college in Slovenia, in his lab in San Francisco. And then I signed up for a bunch of different PhDs. At the end, selected UCSF to continue my study there. But what I wanted to say is that, let's say, I've done bachelor's in Slovenia and I've done PhD in US. I didn't get much experience otherwise. I've never done, let's say, bachelor's in US or PhD in Slovenia. So whatever I'm going to tell you next, it's not going to be apples to apples comparison. But in terms of some, let's say, observations on the positive side, I think Slovenian system gave me a really fantastic knowledge base. So if I compared myself to my US classmates, I could see that I have a lot deeper knowledge when it comes to biochemistry area, or biology, physics, math, chemistry in general. So that was definitely on the positive side. I also didn't have any school debt, like student debt, like many of my colleagues and classmates back there. So it's definitely much cheaper system as well. On the perhaps some downsides, and this is again from 20 years ago when I was doing my college, so things might have changed in Slovenia since then. But what I have missed now in retrospect are that we didn't work on real world problem solving too much. There were some aspects of it, but not as much as I would have benefited from. I think we would benefit, or Slovenian system would benefit from just having students present more, debate, encourage critical thinking, reviewing papers. So not just agreeing with what others saying, but also finding a way to disagree with that. So like really that critical thinking, and perhaps having some flexibility in terms of learning about things slightly outside of our majors. For example, for me, I would really benefit from having a way to learn about coding. So having, let's say, class on computer coding and stuff like that, that would be very beneficial. And at a time, maybe I just didn't know about that as an option, but that would be fantastic to have as well. Another aspect that's perhaps different between the two worlds is more about the mentality. Like in US, especially in the Silicon Valley, where I spent most of my time in, that hustle mentality was just very strong. People worked a lot. They were very driven. And I didn't see nearly as much of that in Slovenia back in the day. So much better work-life balance, I would say.
I completely agree with your points, and I also definitely can relate with studying something outside of the curriculum that the universities gave us, because it was similarly for me after when I did my master's. I came in with a lot of knowledge that I gained during my bachelor's at the University of Ljubljana, and then in my master's I was really feeling like I want to do something else. So I did astrobiology on the side, which was really interesting and definitely not an option at the University of Ljubljana, especially, at least not at the time when I was there. So how and why did you decide to transition to the industry?
Good question. I just always felt like I wanted to be in industry. I never felt like being a professor, teacher. That's probably because I'm a very product-oriented person. I have been bitten by that entrepreneurial bug quite a bit. Like I always wanted to build things. I do enjoy inventing, innovating, prototyping, writing code, doing research quite a bit. But I also like packaging that into some sort of product that others can use. So I was just very excited about that. On more, let's say, operational side, I like to work in teams, organize projects, efforts, talking to customers, users, thinking about resourcing. So just, let's say, more operational processes as well, which I think it's much more appreciated and needed in industry than in academia. It's needed in academia as well, but I guess incentives are not aligned in the same way. So that was just something that I always enjoyed. I never enjoyed grant process. So as a student, as a PhD student, I was a little bit exposed to it. We had to write a few grants and we had to review some grants as well. And never enjoyed that. So didn't want to have some big fraction of my time being spent on that in academia. So we still do some of that in industry, but I do think that the amount of time is much less spent on that as opposed to some other topics that are more enjoyable, to me at least. What I am seeing recently is that there is quite a bit of convergence. I hear many professors are starting their own companies. And if you look at companies, they're doing a lot of research today, especially big tech. They had a lot of interesting projects to work on, some startups today, including ours, as we're going to discuss later. Like research is still core business for the industry. So I think the two worlds are very much intertwined today. But overall, I prefer, let's say, the skills and the type of work that I do in industry. I prefer that over what I imagine I would do in academia. Still, definitely don't want to close any doors because it feels like a nice retirement at some point. But for now, I enjoy industry over academia.
And so after you finish your academic journey, let's say, you went into big tech and worked at Google Health. And after that, you started your own startup. So what made you transition from big tech to a small startup?
Yeah, right after my UCSF studies, I got a job at Verily, which at the time was just spinning outside of Google under Alphabet umbrella. So I spent a few years with them. I was there for nearly 8 years. I was pretty lucky to be there at the golden age or in the golden age of Google, like with all of the perks, benefits, free food, and just an amazing work environment. But what I really appreciated is just being able to work with some of the smartest people that I've ever met. It was really incredible environment, like working with world champions in Sudoku, puzzles, Olympians, and stuff like that. It was just really impressive what sort of people you were able to meet. So as a data scientist in early career, like I was at the time, I don't think there is a better place to be, to learn and grow like big tech, because they have, let's say, fine-tuned the processes for several decades. when it comes to coding, when it comes to good practices, when it comes to optimizing productivity and so on. There's just really so much to learn and gain. So I was really super lucky to be part of that. But yeah, with all the good things that I mentioned, 7 years was enough. So I've decided to quit last year and start my own company. Again, going back to that entrepreneurial bug that I mentioned, that was something that I always wanted to do. Always wanted to have my own company that's gonna do what I enjoy doing, solving problems that I deem impactful. So after spending 7-8 years at Verily, I finally collected the courage. I feel like I have learned a lot in that process to have at least a decent chance at succeeding in my own journey. So here I am. I'm not doing that alone. So that was another nice aspect. So I mentioned all of the smart people. Was super lucky to partner with and have a co-founder, Melissa, who I've worked with at Verily for 3 years. She comes from a business environment. So it's a nice match because in business, let's say, just knowing technical stuff is not enough and just knowing business is not enough. So it's a really nice set of complementary skills that we are both bringing to the table. So yeah, fingers crossed that it all goes well.
So now let's shift towards the topic of today's conversation, and let's start with discussing about proteins. So protein structure prediction is arguably one of the most successful applications of deep learning in computational biology, and it is largely known that the protein's amino acid sequence determines its 3D structure, which it is in turn directly related to its function. That would be chemical reaction catalysis, signal transduction, etc. So the history of protein structure prediction problem goes back to determination of 3D structure of myoglobin by John Kindrew in 1950s, which is a landmark in biochemistry and structural biology. And I'm pretty sure that everyone who studied biology, chemistry, biochemistry has at some point heard about this. And since then, crystallography has been the standard for protein structure determination, and predicting 3D structure of proteins has always been a challenge. And since we have quite a few listeners who maybe don't have biological backgrounds, maybe can you tell us why is knowing 3D structure of proteins important?
Yeah, many reasons. I would probably highlight two major reasons for why I think knowing structure of proteins is important. So one, by studying 3D structure of protein, we just learn a lot about how proteins function, how biology works to the level of atoms, basically. For example, how do enzymes perform their functions? How do proteins bind to each other? How do they bind to small molecules? How do they relay messages from one side of the body to another side of the body, and so on? How do we move? A lot of it is defined by that biology at a molecular level. By knowing all of that, we can learn a lot about nature and use it to our advantage. So, for example, how to overcome problems in protein engineering, and this goes well beyond life sciences. So, agriculture, industrial enzymes, and so on. There is a lot of protein engineering used in those spaces as well. And ultimately, designing novel therapeutic approaches. That's something that we can learn a lot from structure. A really interesting example that I was exposed to early in PhD is, for example, learning how HIV prevents cell-native antiviral mechanisms. There's that famous story about protein named APOBEC 3G. I think there's another Slovenian at UCSF, Matija Peterlin, that contributed a lot to our understanding of that. But yeah, HIV has been a pretty interesting, let's say, model organism about studying protein structures. And there was that protein in HIV proteome called VIV that somehow prevented the functioning of the human protein APOBEC 3G, which is an antiviral protein. It works by introducing hypermutations to viral genomes. So it basically mutates viruses so much they just can't replicate anymore. But HIV found a way to counteract that mechanism, and that's been pretty mysterious for a long time. And studying structure of proteins and protein interactions of this HIV complex, we have finally figured out that the way it does is that it basically brings this APOBEC 3G protein to ubiquity, into the proteome system for degradation. So it degrades the protein that way. And understanding that has really led to a novel therapeutic approaches called molecular glues. So now today, let's say we have many companies, many startups interested in targeted degradation of proteins of interest by creating small molecules, by creating tiny proteins in some cases that bring a protein that we would like to degrade close to the proteasome for degradation. So just as an example of what we can learn by studying structures of proteins. So that's one reason. Second reason is that if we know structures of proteins, we can learn about what sites are important for their functioning, and some of those functions can lead to diseases. So knowing the structure, we can now rationally design small molecules that modulate them in a way that benefits our health, and ultimately becomes a drug, a therapeutic as well. So that's an area that's becoming more and more popular, especially now with AI, ML approaches. I think these are going to have profound impact how well we are at designing small molecules with desired therapeutic properties. So yeah, that's an area that I'm super excited about. I think we are already seeing with AlphaFold and some other approaches how much impact that is having, and this is just the beginning. I think the future is going to be much better than where we are today.
So you already started mentioning about protein structures, and as we said, protein structures can convey a lot of information about their biological processes and molecular function and cellular components. However, there is no direct mapping between these and their functional annotation, and they are still a challenge. Can you maybe expand a little bit more on this?
Yeah, so maybe what you're referring to is that we know how to map between sequence and structure a lot better than we can map between sequence and function of a given protein. And yeah, the reason for that is that, well, the nice thing about structures is that they are very close to the ground truth. So for a given sequence, you usually just you have one option for what the structure of that protein is gonna look like. And yes, proteins move, proteins are flexible, and they can change conformations depending on the signal and stuff like that. But for most of the cases, for the majority of the cases, there's just one structure. So knowing the structure, that gets you as close to the ground truth as possible. But when it comes to the function of proteins, that's not such a straightforward concept. Function is more elusive because proteins, for one, can have multiple functions. They can have different functions in different times. For example, with post-translational modifications, where they are in cells, where they are in our bodies, what sort of phenotypes we are exposed to. Proteins can have different functions in those cases. Defining what a function is, it's not always black and white type of an approach. So there are many gray zones. There are overlaps between different functions. So functions are not well defined, and we still don't know many functions of the protein. So the experimental approaches to map those functions are fairly incomplete. You either have very narrow efforts where we study one specific function for a specific small subset of proteins, or where we go with these high throughput or large scale approaches that are pretty sloppy and making lots of mistakes. So all that I'm trying to say basically is that structures of proteins are a lot better labels, a lot cleaner labels than functions. Functions are just super noisy labels. So any efforts in terms of predicting or mapping sequences to functions are just going to suffer from that noise quite a bit.
And what kind of data sets, so labeled, unlabeled, are used in modeling, and what are the advantages and disadvantages of both?
Yeah, a lot of my research and a lot of, let's say, my work in the last decade and a half has been around applying KIML data science approaches to life science data sets. And what I have learned is that projects or efforts that work with large data sets and high quality data sets, there it's fairly easy to succeed. If those two criteria aren't met, so for example, if the data sets are too small or too low quality, too noisy, just as what we have discussed about functions, then the chances of success will drop down quite a bit. So, and that's a big challenge for life sciences in general, because a lot of efforts are geared towards collecting fairly small data sets. So doing experiments in science is expensive. We can't afford to collect data points on millions of samples. Usually we keep those pretty low. So modeling, analysis, and so on is heavily going to be determined by how large or how small your data sets are. When it comes to quality, what's really, wouldn't call it problematic, but it's definitely a big challenge, is that the data that we are collecting is not very reproducible. So there's a lot of variation depending what instrument someone used, what, let's say, protocols went into experiment design and execution, what phase was the moon in. When I was doing wet lab experiments, I just noticed that, like, even repeating everything in the same way, you might get different results. And that does have consequences. That does have impact on how accurate the models and analysis are going to be down the line. So yeah, when it comes to choosing the data sets to apply to modeling to, it definitely comes down to data sets that are large enough and that are as high quality as possible. And there's not a lot of places like that, but yeah, it's growing rapidly. So again, another aspect that I'm very optimistic about in the future. So just the amount of data sets is always going up.
And now I would also like to talk a little bit about one of your papers. So of course I was looking a little bit at your publications, and I thought that this one is quite interesting, and it is about cryptic sites in proteins. Basically, these cryptic sites in proteins are elusive pockets that remain undetectable until they are triggered by a conformational shift. So, meaning that they are absent in APO, that means ligand-free structures, but they emerge in hollow configurations, and they hold immense potential for expanding our arsenal of drug targets, and especially in proteins with seemingly inaccessible binding pockets. So what defines a cryptic site in proteins, and how does it differ from a traditional binding pocket?
Yeah. So again, going back to the structure of proteins. So the structure of proteins is not static. Protein moves; they change their they change their shapes, so to speak, quite a bit, depending on the protein and what it does. But basically, what defines a cryptic pocket is exactly as you said. So some proteins, in the absence of small molecule ligand or another protein, the site where that small molecule is going to bind to is not well defined, and in some cases that site is pretty much flat, so there is no pocket even to be observed in unbound conformation of the protein. And it's only when small molecule comes by that pocket is being formed, and the small molecule can sit into it or bind to it. It's a really nice property of proteins. The reason we are interested in it is because there are many proteins that seem to be undruggable because they have no visible pockets in their structures. And for some time, we have thought that there is no way to go after those, but now there's a growing set of examples where that's not the case anymore. So there are plenty proteins that don't show like they could bind small molecules, but it's really just about finding the small molecule that's going to open up, that's going to relax the structure a little bit and be able to sit into it.
And what kind of methods are employed to identify those cryptic sites, experimentally and computationally?
Yeah, on the experimental side, the approach that had a lot of success is covalent or fragment binding screening approaches. So basically, that includes having small molecules being screened against a bunch of different proteins and see what proteins they're going to bind to. And then once you have this covalent interaction between protein and small molecule, you can go and figure out not only what proteins a given molecule binds to, but also where, because they are, again, covalent binders. Finding small molecules that bind to proteins serendipitously, and then using X-ray, NMR, and other approaches to figure out how does that happens, that too. But yeah, a lot of, on the experimental side, a lot of these efforts are happen by luck, so to speak. It's really difficult to rationally design and go after these cryptic binding sites, especially because I forgot to mention that earlier, but basically there are two mechanisms for how cryptic sites can form, how cryptic pockets can form. One is just random sampling, so proteins are going to exist in different conformations in different points in time, and maybe some of those conformations are in the right shape to accept a small molecule by, again, just by random sampling. But there are also cases where small molecule induces the shape formation to be able to accommodate for it in the protein structure. And that, let's say, cryptic sites that are formed by induced fit type of mechanism, those are really difficult to study experimentally or computationally for that matter, because you really need to find the right molecule that's going to induce the right changes in the conformation of proteins. And then back to your question, on the computational side, the approaches are, let's say, molecular dynamic simulations. Those are still prevalently used to identify what sort of conformations a protein is going to adopt. And then the AI/ML approaches, like the one that you are referring to by citing the paper I authored a few years ago on cryptic binding sites, that's also a really interesting avenue to go after finding cryptic binding sites. It turns out there is quite a bit of biological information stored in proteins. So as part of that project, we have developed, let's say, a machine learning algorithm that took different features or descriptors of protein structures and sequence as an input and predicted whether a site on a given protein contains or is part of a cryptic binding site or not. And it turns out features like how flexible a given site is, as let's say approximated by molecular dynamic simulations, was the most important features. But the next most important feature was evolutionary conservation. So it seems like these cryptic sites are a big part of biology on its own because they seem to be quite conserved evolutionary as well. So just an interesting observation about that.
Very interesting. And what impact did the prediction of cryptic sites have on the druggable human proteome?
Yeah, I think the biggest impact that we had was just to, not to demonstrate, but hint at druggable space being a lot larger than what we previously thought. So previous estimates were that a very small fraction of proteins can bind drug-like small molecules. Analysis that we have done, and many people afterwards suggest that that's not the case. We can go after proteins that don't seem to have any pockets whatsoever. It's just about finding the right molecule that will induce changes that we need for them to bind to.
And now we are coming to an interesting topic, which is talking about your startup. What is TesorAI, and what is the goal of the products, and what are the plans for the future?
So maybe to set the stage first, we spoke a lot about proteins, protein structures, drug discovery. So this is a very important aspect and factor in finding and developing new drugs. But there is also another aspect when it comes to these sort of efforts. And those efforts include being able to find what a good target is, what is a good biomarker to diagnose someone with disease or predict outcomes to treatment, predict prognosis, and so on. So that there are many aspects that involve deeper dives into biology itself. So let's say drug discovery that we have discussed so far through protein structures, finding small molecules, cryptic binding sites, and so on, that's very small molecule centric. It doesn't take bigger organism into account. So let's say, are there going to be side effects to given treatment? Or what is the best target to begin with? It's going to minimize the side effects, but maximize the efficacy or the desired impact. That's a very important aspect that goes into drug discovery as well. And outside of that, in more clinical or healthcare world, biomarkers are a very important aspect in terms of identifying diseases very early, identifying what sort of treatment is going to work best for a given patient. So that whole notion of personalized medicine leans a lot on finding the best biomarkers for given diseases and treatment. So and at the end, is the drug effective or not? That also requires some sort of measurements of the phenotypes. So a lot of that work we haven't discussed, but it's just as important, if not more important, than that rationally drug-centric approaches that we have mentioned so far. One quote that maybe just speaks to how important it is to find the right drug targets. So a lot of people who have spent decades in drug discovery, they've been telling me that having a bad drug against a good target is a lot better than having the best possible drug, the best possible small molecule against the bad target. So it just speaks to how important those, let's say, steps before or after drug discovery are. And what's really interesting is that genomics, DNA sequencing, let's say in early 2000s, have been very exciting, promising area for how to find new targets, how to diagnose people, or how to predict whether somebody's going to have a disease or not. But it turns out that didn't really quite live to its promise. DNA or genomics-based approaches still have many challenges. And it turns out a lot of life science biotech industry suffers from the same challenges. I, as a computational biologist, have encountered the same challenges. And I would group them into three categories. The first one is data integration. So just as mentioned, genomics provides one level of information, but there are many other aspects when it comes to biology, from RNA, transcriptomics, proteomics. Proteins we've discussed quite a bit are super important in that space. metabolites, so metabolomics, all of these area contributes to the puzzle. And a big challenge is how do we combine all of these signal so they don't just add up but multiply from each other. So data integration is something that we haven't been able to solve for very well just yet. Second one is what I call fat small data sets. So as mentioned, a lot of life science is based on very expensive experiments. A lot of our efforts are going into collecting as many data points per sample as possible. So as many mutations, let's quantify as many proteins as possible. But because it's so expensive, we can only do that on a small number of samples, on a small number of patients or participants, so to speak. And from AI ML perspective, that's a really difficult, really challenging space to be in, because normally we would want the opposite. We would want to have fewer number of features per sample, but as many samples as possible, millions, tens of millions, hundreds of millions, to be able to have some success in that space. So just thinking about, let's say, approaches that are going to work in life sciences, we do need to take these fat, small data sets into account. And then the third category is what I call data or information loss. So a lot of data goes through multiple stages today. So it's first collected by all of those expensive instruments in the lab in the form of raw data. But raw data is not something that we analyze at the end. That raw data goes through pre-processing steps, it goes through filtering steps, it goes through aggregation steps, and so on. So there's a lot of modifications that happens along the way. And what I have noticed is that most of the efforts in computational biology or data science are being spent on steps after all of those that I have just mentioned. All of these pre-processing steps are very much overlooked. And because they are overlooked, when you look at the algorithms running or powering these pre-processing steps, those are still fairly simple. Many of them still only work on Windows workstations. And what's under the hood is just very simple steps. And for example, in mass spectrometry proteomics, we today have about 80% of spectra that remain unidentified. So we only are able to identify 20% of analytes that are actually in the sample. And you can imagine how that's going to impact the success rates downstream when you have 80% of data that we can't even touch on because of these algorithms that are being pretty inefficient today. So I gave you a very large and high-level overview of the problems that I believe computational approaches or AI approaches can have an enormous impact on. And this is what we would like to address with TesorAI. We are starting with this information data loss, so the last category, the third category that I mentioned, just because it seemed to be the easiest step to get into this space. And there our approach really is to start migrating these simple algorithms from workstations, again, in many cases, Windows workstations, onto cloud. And this is one going to solve the problem about speed. I didn't mention that, but many pre-processing steps are much slower today than what it takes, the time that it takes to generate the data. So they are a bottleneck in the lab, so to speak. But we can speed it up by migrating to cloud. And now that we are in cloud, we have access to a lot more compute. So we can start replacing these simple algorithms with a lot more advanced ones. And our early products in mass spectrometry proteomics today, we are already demonstrating that we can recover 4 times or more as much of analytes than standard algorithms out there. So imagine being data scientist, computational biologist, all of a sudden, just using the same data that you've been using yesterday, you can now work with 4 times as many proteins in your data set. I think that this is really going to change the success rates that one is having with their experiments, with their data. So that's in a nutshell what we do.
Oh, amazing. So you briefly mentioned one of your products, but you are currently offering 2, if I remember correctly. So may you just expand a little bit on the second product?
Yeah, the first product that we call TesorAI Search is all about being able to see more of what's in the data that teams have been working really hard to generate on. But that doesn't solve all of the problems that we as a biotech industry community are having. So seeing more is not necessarily the final solution, because in many cases, analytes in experiments are not all biologically relevant. Some, or in some cases, many of them are artifacts from experiments. Think of it as contaminants, artifacts, and so on, or let's say some sort of artifacts from how we've designed experiments and so on. So it's really important to know what sort of hits, what sort of analytes that we are seeing in experiments are biologically relevant versus what's just some sort of noise. And for that reason, we have created this second product that we call Refine, that basically is an integrative approach that basically combines experimental measurements with our knowledge of proteins and small molecules to reduce those false positives in high throughput assays in particular. So any lab that's collecting massive amounts of data, they are very much struggling with false discovery, both false positives and false negatives. And this Refine method or product that we are developing helps them use any information that they have collected through experiments, but also properties about proteins and small molecules through foundation models that we are building to help select for those that are the most biologically relevant.
And where are you getting the data to train the models?
Through a couple sources. One is publicly available sources and through our partners. We are not generating data of our own yet because the amounts of data, I think, so going back to what matters in AI ML or analysis and modeling, it's really data set sizes and quality. For the approaches, for the modern approaches in AI ML, that's going to require tons of data. And generating tons of data yourself is just very expensive. It's not very efficient. Plus, you would have many biases because you would always generate data just in a certain way. So if you develop a model that's based on data from a single source, that model is likely not going to generalize very well. So in this space, in this industry, it's very important to build models based on data that come from different sources, because those are going to, maybe they're not going to have as high accuracy overall, but they're going to generalize a lot better to a new data source afterwards.
Do you think that is a bit of a downside in academic computational biology, not having the partners who would provide you data sets?
I don't think so. In industry, it's a lot more difficult to get access to data from different places because that's the value that they're having. So I think, if anything, being in academia might be an advantage because academic institutions or scientists, academic scientists, are a lot more open to sharing the data. Whenever they publish the paper, many papers, many journals today require them to deposit raw data into public repositories for anyone to see. So yeah, I think that might be even an advantage of being in academic space, just having more access to different labs and resources in that way.
Okay, and now I have some questions about emerging trends and later on a little bit about the challenges of the field. So, what are the emerging trends and the future direction in computational biology that are bound to make a significant impact on the healthcare in the coming years?
Yeah, I've mentioned it a few times. From my perspective, what I know about AI/ML, it's already making major impact, and I do think that we are just at the beginning of it. That field is going to have a lot more impact every year. So um, there's no question. Like this is really going to change how we think about biology, life sciences.
Is there anything that you are particularly excited about?
Yeah, I like to think about, so when it comes to AI/ML or using AI/ML to solve problems in biology and life sciences, I like to think about it in an end-to-end way. So a lot of things that we are doing today are still fragmenting, breaking apart problems into smaller problems, and then trying to solve for that one. For example, in drug discovery, rational drug discovery that we have discussed, solving protein structure is an example of a smaller problem, an important problem, but it's just a small problem. Like the next step is let's find the right molecule that binds to it. And then you have to figure out whether that molecule is toxic or not by binding to other proteins, optimized for affinity, optimized for shelf life and stuff like that. So there's many other problems down the line that we have to optimize for. So I think with more data being collected, with better approaches in AI/ML being built and invented, innovated, the future is gonna move towards more end-to-end solutions, so more holistic approaches. So let's skip all the layers in between. Let's not think about smaller problems in between, but let's think about it everything, like the whole thing, end to end. For example, um, what is the disease that I might be having from just a drop of blood? So not just looking at one disease, but can we predict many diseases at the same time that we might be having at the moment? Or if we have a compound, not just being able to predict what proteins or how it binds to a given protein, but does it actually induce the right phenotype? So does it have the right desired effect, and does it not have undesired effects, and so on? So that just going all the way to the end might have better performance in the future. And let's say if we are going to be super successful, then we can just name a disease, and a computer is going to generate a small molecule that's going to become, let's say, an instant drug altogether. So yeah, again, what I'm excited about are these holistic end-to-end approaches, because by breaking problems into smaller sub-problems, we are losing information. Like there is information by taking, let's say, our knowledge all the way through the end. And that's something that we are not doing yet, but I think we are going to be seeing more and more of that.
And what are some of the major challenges in the field?
Data set sizes and quality. That is, these two are always the most important aspects of the work that we do. New approaches in terms of how we encode certain biological data or any data, so that the models can, or that computers can work with it. That's also an important aspect. So a lot of innovations are probably going to come from that space as well. So not just data set sizes and qualities, but also model architectures, so to speak, like transformers in language modeling, for example, were super successful for that problem. I think we need an equivalent for that for biology as well. So what's going to be the right architecture that's going to capture information about biology from the data that we are collecting? That is a challenge as well.
Yeah, absolutely. And before we turn a little bit to more philosophical part of this podcast, we just a little quick question. How do you see Slovenia and Slovenia's startup space fitting in the computational biology? Do you see any startups popping up in Slovenia in this environment?
Good question. Yes, I have moved back to Slovenia, at least temporarily. But the startup, so my co-founder, she's still in USA. So the startup is still mostly US based. We do have some employees now throughout Europe and in different places in US and elsewhere in the world as well. So we are more of a global virtual company already as is. But yeah, haven't dove too deep into Slovenian environment yet. What I do see is that we have amazing talent in Slovenia as well. Like I have several colleagues at Alphabet, Google, who are doing amazing work. Other big tech industry, like there are some fantastic people that come from there as well. And even talking to, let's say, some people from Slovenia that responded to pings or reached out to me, like the talent is definitely there. I haven't been too much in the, let's say, in the startup ecosystem, so can't really speak to that unfortunately yet, but hoping to do more of that.
As promised before, now we are diving a little bit into philosophy, let's say, or at least more, let's say, ethical implications. So, as our understanding of the human genome and proteome grows, what are the ethical considerations and considerations surrounding genetic engineering, gene editing, and personalized medicine? How do we balance the benefits of these technologies with concerns, let's say, about privacy, equity, and unintended consequences?
Yeah, I just want to preempt this by saying that I'm not an expert in these areas. That's not something that I'm thinking much about. It's an important aspect, so yeah, again, don't get me wrong, but yeah, not something that I've been diving and thinking deeply about, but certainly important aspects. And like I do think that, let's say, these equalities is going to be a big thing to be discussed about, because let's say as we develop new approaches, new therapeutic approaches, as we think about, as we understand how biology works better, like whether someone is more susceptible to having a disease than other, like just what sort of implications that's going to have from health care, health insurance, and so on. Yeah, certainly very important aspects to think about. Does our biology define who we are, how we act, how we respond to certain things? That, yeah, I don't think there is, let's say, a free will that we have. I think a lot of it are set by biology, not only by biology, but also by So not only the internal biology, but also external signal that we are getting through environments by interacting with others, by interacting with nature, by things that happen to us all the time. Yeah, the way I think about it is like there is genetic code, or if I draw some parallels to computer science, you have code and you have compiler. So you need to have a system that understands the code and runs it. And when it comes to humans and our biology, we have genetic code as well, which is similar to computer science code. But we also have compilers. So when we are born, like just knowing your DNA is not enough to learn about what's going to happen at the biology level. It's also important to know what sort of proteins did you have in your first cell, where were those proteins located, what sort of post-translational modifications did they have, what were the cells around it, what sort of signal did they start collecting very, very early on. Unlike, let's say, computer science compiler, I think our compilers keep evolving. So as soon as we receive feedback, that our biology compiler gets updated along the way as well. So yeah, I do think that things are complex. I'm not sure if anybody understands it well enough, but yeah, there are so many. So like it's a loaded question, and there are so many areas that are really, really critical for the future, especially with these new technologies that are not only getting better, but coming in here faster and faster. Let's say the speed of evolution is just ramping up as well.
It is, in fact, a really loaded question. And another question that I would like to ask, since you were in Silicon Valley, and I'm sure there have some people in the Silicon Valley who are very into trying to reverse age themselves. How much control do we have over our biological destiny?
um, I think quite a bit. When it comes to age or when it comes to health, yeah, there are quite a few things that we can do. Again, not an expert in that area, but what I'm hearing through or what I'm reading about, yeah, what we eat, diet, how much do we exercise, do we surround ourselves with positive, optimistic people, like are we social beings and so on, like that all, there's pretty solid evidence that that does improve our health, short and long term. In terms of pushing the limits to live much longer than what we've been seeing so far, I think that's going to require a lot more work beyond just better diet, exercise, and so on. I don't see a good reason for us not being able to get there, but yeah, it's very, very early days, and biology is complex. Like what's really interesting is that with many animal model organisms, we were able to extend their lifespans quite a bit. There are many cases from yeast, I think even mice or rats and stuff like that. We were able to extend their lifespans significantly, in some cases multiples of factors. So, and if we can do that on smaller or smaller models, like why wouldn't some of that replicate and transfer onto humans? I don't see a good reason. Yes, we are a lot more complex. We are a lot bigger beings with a lot more, like the bigger you are, the more random effects are going to happen to you, random mutations in different parts of our bodies that are very difficult to fix specifically. So yes, I do think that it's going to be very challenging, but yeah, I like a few years ago, I wouldn't think that, let's say, these cell therapies or genetic gene editing and so on, like that with CRISPR, that has changed what we are capable of very rapidly. And yeah, sometimes things seem flat at the moment, and it just takes one breakthrough to look at what's possible, what's feasible from a very different perspective again. So yeah, I think our progress in general is not growing linearly. It's very step-like function. So we are seeing CRISPR is having big impact, and it's going to have bigger impact as we move along. The same for AI, and it just takes, like, there's probably going to be many examples of these sort of breakthroughs in the future. So yeah, I'm optimist by nature, as you see. So I do think that we can change a lot of things. Also, what's very interesting to me are all of these brain machine interfaces, BMIs, with Neuralink and many other companies. We as humanity are finding ways to connect with ourselves, with our biology, with our brain at a very different level to what's been possible so far. So our future might not be in biological matter. Like we could just move ourselves into silicon. So who knows?
That would be interesting. Okay, so we came to the end of our conversation, And maybe for a last question, I would like to ask you if you have any recommendation for books for our listeners that would be interested in delving deeper in the topics that we discussed today.
Ooh, I'm not reading too many books about biology and AI. I mostly go for papers, scientific papers right away. I read Jennifer Doudna's biography that came out recently, I think about a year ago. I think it's called Gene Breaker or something. Don't quote me on that. I'm bad with names. So that's an interesting space to be. I think the AI ML is just moving so quickly that people who are writing books can't, like it's difficult to keep up with all of the discoveries. So there's probably not so many books that would be super relevant, or once they are published, they're already old. What I am reading usually are just lots of biographies. I'm very much into reading about how did other people succeed, or what problems did they have to deal with. So let's say for any young people, I'm trying to get that to my son as well, who's 12 or is going to be 12 soon, and he's been reading lots of biographies with me as well. But yeah, that's what I would recommend to many young, or anyone basically. Like biographies, they have so much wisdom in them. It's pretty good.
Yeah, absolutely. I completely agree. And I just remembered one really good book that I read quite recently, and it's titled Deep Medicine from Eric Topol. And it is a book not specifically about computational biology, but there is a chapter about computational biology, also computational chemistry, and it mentions a lot of different professors who are working in this space. So
like maybe another, like to make a whole circle and go back to the first question on academia versus industry. Like the books that I have enjoyed, there are a few books about drug discovery, not only about the science of it, but also about people and politics and so on, and how difficult it is to push drugs onto the market. Again, not just from scientific clinical perspective, but also dealing with these large organizations of pharmaceutical companies and so on. But yeah, the books that I would recommend from that perspective are HER2, basically on how Genentech developed Herceptin, the breast cancer treatment that's been very successful, or $1 billion drug is also another very interesting book about not just about science of making a drug, but also dealing with people and organizations and regulatory aspects and so on. So it gives very different perspective to scientists also in terms of what one needs to deal with when it comes to real world scenarios or situations.
Indeed, especially that $1 billion drug. Amazing book. Thank you again, Peter, for that really wonderful conversation. Wish you all the best with your startup.
Thank you so much as well. Thank you for inviting me.